Hello, Jessa. It's Dr. Greenberg here. This video message may be coming as a surprise to you, especially after we had such a positive conversation at our last meeting. But my coworkers and I decided to take a second look at your files, and we're sad to tell you that you have stage 4 pancreatic cancer. This just means that the cancer we thought was non-existent has unfortunately spread from beyond the pancreas to nearby blood vessels and nerves. At this stage, it's considered metastatic, and I'm sure this is confusing because we told you that everything was heading in a good direction and that your symptoms were minor. But let me just explain to you how we diagnosed it and how this occurred. The cell goes through a cycle of stages. The main two stages are called interphase and mitosis. The interphase takes up a majority of the cell's life and is the preparatory stage for mitosis. In interphase, the cell grows and metabolizes. The first phase of interphase is G1, or the gap1 phase. In this section, the cell increases in size, and it is the preparatory phase before the S phase. And the S phase is just the DNA replication phase. This allows for each cell created by cell division to have the same genetic makeup, and DNA is a type of nucleic acid. It's a monomer is a nucleotide, and DNA is double-stranded and uses base pairing. DNA is made up of three main components: phosphate, deoxyribose sugar, and a nitrogen base. The nitrogen base can be a purine or a pyrimidine. Purines consist of adenine, guanine, while pyrimidines consist of thymine and cytosine. DNA undergoes semi-conservative replication, which means that each half of the original DNA molecule is joined with a new complementary DNA strand. After the S phase is the G2 phase, or the gap2 phase, where the cell continues to grow and prepare for mitosis. Now, mitosis consists of 5 stages: prophase, prometaphase, metaphase, anaphase, and telophase. Mitosis is the actual division of the cell, and it's the process in which one cell becomes two. In prophase, the centrioles move to either side of the cell, and the DNA condenses into chromosomes. In prometaphase, centrioles start to make fibers called microtubules. Microtubules start reaching out towards the chromosomes, and in the metaphase, microtubules attach to the chromosomes and make them align in the middle of the cell. In anaphase, microtubules pull chromosomes apart and towards the microtubules. In the last phase, which is telophase, those microtubules disappear and separate and cause two nuclei to form. The cell starts to pinch and divides into two. Now, sometimes things can go wrong in the cell cycle. In the synthesis phase, which is in the interphase, many things can go wrong. When the DNA replicates, there can be an error because it's an exact copy of each strand. Since there are several copies created, there can easily be an error. Cells go through different checkpoints to make sure that they are ready for the next stage of the cell cycle. However, some of the cells that aren't ready are sometimes able to pass through the next stage, even though it leads to complications. Cancer is an abnormal and uncontrollable growth of cells. It's caused by mutations of the cell, and an unhealthy lifestyle such as smoking, not exercising, and even sometimes genetics can allow these mutations to occur and eventually cause cancer. I just wanted to say that I am sorry I had to break this news to you and your family over video and not in person. Please come to see me as soon as possible and take this time to process and understand this information. I'll be contacting you in the next few days to schedule our next appointments. Again, I'm very sorry to break this news to you, and I'll see you soon.